Hi everyone, as Kelly said, my name is Hector Garcia Martin and I'm the Host Engineering Deputy Director in the Joint BioEnergy Institute. Today I'm going to be talking about metabolic flux analysis of biodiesel-producing E-COLI. The content of my talk is listed here. I will give you a brief intro to JBA, to the fuel synthesis division, and to the Host Engineering Director, which is mine, and explain you what kind of things we're doing there. Then I'll give you a brief introduction to flux analysis, which is the main tool in computational host engineering at JBA. I will explain the two most popular techniques, flux balance analysis and 13C metabolic flux analysis. And then I will explain our efforts in creating a fusion of these both techniques that will have the benefits of both of them. And then in order to show you how these techniques can actually be useful for metabolic engineering, I'll give you a working example of biodiesel-producing E-COLI. I'll show you how the production of the fatty acid ethyl ester changes the E-COLI metabolism. I'll show you how we develop a method to determine limiting factors, and a limiting factor is the answer to this question. What does my strain need to produce more biodiesel or more X of a given product? And then I'll talk a little bit about some knockout predictions that we made in order to increase biodiesel production. And finally, I will very briefly mention that we're doing similar work on yeast, and we're trying to use 13C metabolic flux analysis for microbial communities as well. Let me start with a brief introduction to JBA. This over here is JBA in Emeryville. Is the product of a $134 million grant by the DOE for 5 years. It's 1 out of 3 bioenergy research centers. It started its operations in 2008, and it has now around 200 employees from 6 different institutions with different capabilities and strengths, including the Berkeley National Lab, UC Berkeley, Sandia National Lab, UC Davis, Lawrence Livermore National Lab, and Carnegie Institution for Science. Now, as you probably know, our mission is basically very similar to yours. We are here to produce cost-effective, energy-efficient, and commercially viable lignocellulosic fuels. This is our work chain. We basically grow plants. We take the energy of the sun, we store it in plants. We take that lignocellulose, we deconstruct that through pretreatment and enzymatic treatment into monomers, and then we ferment those sugars into biofuels with the aid of genetically engineered hosts, in our case E-COLI and yeast, and we're working also on sulfolobus. For each of these processes, we have a division. The feedstocks division, the people in the feedstocks division are in charge of doing the genetic engineering to produce plants that are easier to deconstruct and drought resistant and have the qualities we're looking for. The people in deconstruction are in charge of pretreatment and developing the enzymes necessary for the most efficient deconstruction. And finally, we at Fuel Synthesis are in charge of doing the fermentation from sugars into biofuels. We have in Fuel Synthesis 5 different directorates. Each of them has a different mission. The Biofuel Pathways Directorate is led by Harry Beller, and he basically looks in exotic places for life, for bacteria that does interesting stuff to create X. X is typically a biofuel in our case, maybe diesel or a gasoline molecule or any kind of fuel molecule. Then he takes the DNA that contains the pathway that produces that X, and they introduce it in the host, either E-COLI or yeast. The people in metabolic engineering, led by Tek Soon, are in charge of tweaking this pathway. They change RBSs, they change promoters in order to suck as much carbon from the central carbon metabolism into that pathway. We at the host engineering are in charge of meddling with the rest of the organism, or the metabolism of the organism, in order to push as much carbon into the pathway. We do that by typically, or one way we do these things is by doing knockouts of the genes that are taking carbon away from our pathway. But that's just one of the possibilities. Then the people in fuels toxicity are in charge of eliminating X, because X is in many cases toxic to the cell, and it doesn't let it grow as much as we would like it to. So one of the things they do is they express pumps that will take this metabolite out so growth can continue uninhibited. And then finally, the people in synthetic biology, led by Nathan Hilson, are in charge of automating as much of this process as possible. And one of the things they created is JFI, in which you can create, which is an automated way to create your and to design and create your own pathways. Now, the project that I'm going to be talking about is encapsulated in host engineering. There are two directors in host engineering. Swap is in charge of the more experimental combinatorial approach to host engineering, and I'm doing the computational side of things, which is based on flux analysis. Now, flux analysis, what is flux analysis? Well, this over here is central carbon metabolism for E-COLI. We have glucose coming in. You can see the pentose phosphate pathway that is producing a reducing equivalent, the glycolysis producing ATP, and so on. This is all of the reactions that will take glucose all the way to our biofuel. And the fluxes are the number of molecules per unit time that go through each of these reactions, measured in millimolar per grams of dry weight per hour. Now, why do we care about this? Why bother about fluxes? Basically, because it's our most relevant phenotype. Transcripts and protein expression profiles are important, but in the end, what we really care about, what we're trying to optimize and maximize, is our fluxes. The biodiesel that's being produced is a flux, is the product of a reaction that takes the ethanol and the silicon-CoAs all the way into the fatty acid ethyl esters. And growth rate is also a flux, is the amount of carbon that is devoted to creating a new cell. So we are interested in dealing with these fluxes. I will now explain a little bit, very briefly, what are the main techniques that are being used: flux balance analysis and 30C metabolic flux analysis, and I'll introduce the kind of fusion of these techniques that we're trying to do. Flux balance analysis starts with a metabolic reconstruction, where you take a genome, you look at all of the metabolism into that genome. In the case of E-COLI, the standard model is the ECJR904, which has 904 reactions and 625 metabolites. And the kind of information you get out of that is, for each reaction, you get the reactants and the products. Now, you have one of the reactions that you have is the biomass. So over here is the reaction that tells you all that goes into biomass to create a new cell. Now, that by itself is not enough information, so you use some measured fluxes as constraints. You measure at point 0 and at point 4 what is the glucose that you have in your media, and you can derive how much flux of glucose came into your cell in that moment. The same way you can do that with, let's say, pyruvate of acetate, and that will tell you what is the flux of pyruvate coming out. So these are you're going to use as constraints, but there are still a lot of fluxes that are left indeterminate within those constraints. What you do, in order to find a flux profile that is what you expect to find in your cell, you find a flux profile that will maximize your growth rate. You will assume that your cell has been optimized for maximum growth rate or maximum fitness, and you will look all over the flux profiles to find the one that gives you the biggest amount of biomass. And you will assume that is the fluxes that you'll find in your cell. Now, this, of course, is a big assumption. Many people are not particularly happy with it, and that's why we're doing something else on top of that, which is called 30C, metabolic flux analysis. Now, in spite of that, it's been a very simple approach to try to determine the fluxes inside your cell, your system. It's been incredibly successful. It's been used to improve the production of succinic acid, lactic acid, lycopene, valine, ethanol in E-COLI and yeast. And the way that is being done is through the use of some very simple mathematical constructs. I will explain you a little bit of MoMA, because that's something that we'll be using in the rest of my talk too. So, so far in the previous slides, I told you how people try to predict the fluxes you'll find in a given cell. Now I'll explain to you how you can use this method to try to find the knockouts that will increase the amount of, let's say, biofuel that you're trying to produce. And this method is called MoMA, which is an acronym for the minimization of metabolic adjustment. And what you do in this case, well, before I explain the method, I should explain how we actually view our flux profiles. Imagine that you have a flux map like the one that I showed you before, and we want to have an idea of how to represent that. In order to do that, we use something called the phase space. The phase space is a space which such that for each of your axes, you assign a given flux. So this V9 will be assigned this flux, and this V11 will be assigned this flux. Now, for each of your reactions, or your 900 reactions, you will have a different axis. So you'll have a total of 900 different axes. And one point in this space, like let's say the point over here, will give you all the information for all of the fluxes. So a point here will have, let's say, 80% in V9, 40% on V11, and all the other coordinates will give you the exact description of fluxes for your organism. Now, this green area here, that's the area that is within your constraints, the allowable area. So your fluxes can only be in this green area. If you are over here, that means that your glucose uptake is more than measured, or your pyruvate excretion is more than measured, or it doesn't justify it doesn't comply with the stoichiometric constraints. Now, how do we do predictions with that? Well, in general, flux balance analysis assumes that you have a maximum growth rate. So if growth rate increases in this direction, the fluxes that you will find will be corresponding to this point that has the maximum growth rate within your constraints. If we make a knockout, we are imposing extra constraints. So if we are making this glycosylate shunt reaction 0, we're putting extra constraints. So the allowable area now will be much, much smaller. Now, what will be the flux profile that we will see once we do the knockout and we impose these extra constraints? Well, according to flux balance analysis, it will be the one that has maximum growth rate, which will be this point. But it seems unreasonable that once you make a knockout, your strain is able to adapt and evolve to produce maximum growth rate. So Church and Segre introduced the MoMA principle, in which the flux profile that we will find will be the closest to the initial one within your new constraints. So instead of having the one with maximum growth rate, is the one that is closest to the one we initially have, but within the constraints that we have imposed to the knockout. And this is one of the ways that people are making predictions to this. So what you do is computationally, you knock out every single gene, or couples of genes, or triples of genes in a genome, and see what the result of the fluxes are. One of those fluxes is going to be your fluxes into biofuels. So you can see what is the amount of fluxes into your biofuel you obtain, and compare to see which one is the one that gives you the biggest boost in your production. Now, as I said before, flux balance analysis is incredibly useful, it's incredibly simple, and it's incredibly successful in spite of being incredibly simple. But the growth rate maximization assumption has been questioned in many situations. And so that's why I particularly, the people who decided to try to get a little bit more data to construct your fluxes more efficiently. And that's what we do with 13C metabolic flux analysis, in which we take our cell, let's say E-COLI, we feed it glucose, but not normal glucose, glucose that has been labeled at certain carbons. So some of the carbons, instead of being C12, are 13C. And then we let it grow. We see how we wait for it to spread, for this labeling to spread all over the metabolism until it accumulates at the endpoints, which are going to be biomass, or in particular, or part of them is going to be the protogenic amino acids, of which you're going to have a huge percentage of. At that point, in the middle of the exponential phase, we take a sample, and then we look at the labeling pattern for each of the amino acids. For glutamate, for example, the amino acid labeling pattern will look like this. And this will be the fraction of molecules that have one incorporated labeled carbon, 2 incorporated labeled carbons, and so on. Now, this labeling pattern is going to depend a lot on what the fluxes are going to be inside your cell. So they're incredibly sensitive to those fluxes. The problem is, of course, how to go from this amino acid labeling pattern information all the way to what you want, which are the fluxes. Now, this is what we call an inverse problem. The direct problem will be to go from fluxes into amino acid labeling pattern, which we know how to do. The way that we solve this inverse problem is very standard in a recursive fashion, where we choose a set of fluxes. Then we predict what the amino acid labeling will be for those fluxes. And we can do that because we have what we call the carbon transition information for all of the reactions. So for oxaloacetate formation, we have some coming from the malate dehydrogenase and some of them coming from pyruvate methyl phosphate. And there's some reactions that take oxaloacetate away. Now, for these two reactions, taking carbon into oxaloacetate, we know what we call the carbon reactions. So this is the reactant, this is the product, and this over here will tell you what is the order for the carbons in that reaction. So you know that the first carbon of phosphoryl pyruvate will be the first carbon of oxaloacetate, and so on. And the second carbon will be the second carbon, and so on. If you had more reactions, more reactants and products, you have a similar mapping. Once you have that mapping, what you're interested in getting here is the labeling. right? Which is, again, the fraction of molecules with 1, 2, or 3 incorporated labeled carbons. And the way that we obtain that is very simply adding the contribution from malate plus the contribution of phosphoryl pyruvate, and then weighing them by the amount of relative flux that is coming in. In this case, 0.15 divided by the total of 0.3 plus 0.15, and in this case, 0.3 plus divided by the total contribution of 0.3 plus 0.15. So this is a very simple example. Once you do it for the whole network, it gets more complicated. I'm not going to get into the math of it, but suffice it to say that these Fs over here represent the metabolite labeling, and these EMMs and these summatories here are representing the carbon transitions. But you can find the amino acid labeling for all your amino acids once you know the flux profile. Then, once you have the prediction for the amino acid labeling, you compare that with your experimental results. And we do that through this function called the objective function, where you take the experimental results, you subtract the predicted results, and divide by the error over here. And you keep doing this until your experimental results and your computational results are within your error. And from there, you get your 13C metabolic flux analysis fluxes. Now, both methods, 13C and flux balance analysis, have advantages and disadvantages. In 13C, you can have a more accurate determination of fluxes because you're using much more data. Instead of using just the fluxes that have been measured and stoichiometric constraints, you're using the constraints that are created by the amino acid labeling. That constrains the fluxes from central carbon metabolism incredibly well. So it gives you a very accurate determination of fluxes. Problem is that, and you don't have to assume any growth maximization assumption. You're just trying to fit your data as close to the experimental data as possible. Problem is that usually these calculations are kind of cumbersome, and you can't do it for the kind of large-scale metabolic models that you find in flux balance analysis. Typically, here we work with 80, 90 reactions instead of the 900 reactions that people typically work with in flux balance analysis. Also, we don't have predicted methods for 13C metabolic flux analysis. It doesn't mean they couldn't be developed, but right now there is no standard method that can be used for prediction of fluxes once you have a measured set of fluxes. And finally, 13C metabolic flux analysis does not do metabolism-wide cofactor balancing as does flux balance analysis because we're not using the whole metabolic network. Now, after working with these two methods for a while, I finally decided that I just couldn't do it anymore. We have to combine them or fuse them somehow. So, wouldn't it be nice to have a fusion that has the advantages of both of them? And that's exactly what I'm doing. But I'm doing in a peculiar way so we can take advantage of both methods without having their inconveniences. So I'm fusing 13C metabolic flux analysis and flux balance analysis in the following way. Imagine all of these are all of the reactions for a flux balance analysis models. What I'm doing right now is I'm tracking metabolic balances for the whole genome, and then I'm doing carbon labeling tracking only for central carbon metabolism. Because the fluxes, the amino acid labeling is going to be mainly determined by the fluxes in the central carbon metabolism. We don't need to track how the carbon travels from the rest of the metabolism. And that's equally true because most of the carbon is flowing away from central carbon metabolism. Now, this is the method I'm working on. It's finished. I'm looking at the data at this very moment. I was trying to present it here, but in the end, there's a few other things I had to do, and I wasn't able to finish that. So I'm going to present my previous results from a temporal solution that tries to achieve the same result, which is, okay, I have my 13C metabolic flux analysis for, let's say, the central carbon metabolism. This is glucose, this is pentose phosphate, and I trust them quite very much. So what I will do to give me an idea of whether this will give you the results that I'm looking for is take these results and then constrain my flux balance analysis my flux balance analysis problem with those constraints. So I'll constrain the glucose 6-phosphate dehydrogenase to be 0.67 plus minus a little bit, and so on. So I will look for the maximum growth rate within the constraints provided by the 13C metabolic flux analysis. Now, the idea of this is to give you, so far my presentation has been fairly abstract and more theoretical. But I'd like to convince you that these kind of methods have direct use in metabolic engineering. So I'm going to use this temporal solution to show you the kind of things that you can do with flux analysis and 13C metabolic flux analysis in order to do metabolic engineering on a day-by-day basis. I will give you an example with a biodiesel-producing strain, and I'll show you how the fatty acid ester production changes the E-coli metabolism. I will show you a method to determine these limiting factors, and I'll show you a few of the knockout predictions. Now, this is the strain that I got for metabolic engineering. It was developed by Eric Steen, Greg Bokinski, and Connie Kang. And they basically developed a 3 plasmid system that will take hemicellulose, will convert that into cellulose, that will go into glycolysis, producing one of the plasmids will produce the acyl-CoA, the other one will produce ethanol, and the last one, the thioesteres, will combine them together to create fatty acid ethyl ester, which is a biodiesel molecule. Okay? So this is the system that I was studying with this method. The only difference is that instead of using hemicellulose, I was using labeled glucose that goes into glycolysis from the very beginning. Now, the goals of this project was, one, to measure these flux profiles of the wild type and biodiesel and understand what kind of burden the creation of fatty acid ethyl ester is creating on the cell metabolism. And the second one, more practical, is to try to predict genetic manipulations that would improve this biodiesel production. So I started with my 13C metabolic flux analysis fluxes, measured for both of them, and then, as I said, I took them as constraints into the flux balance analysis model, and I saw within those constraints, within the constraints of the fluxes measured through 13C, what is the fluxes that are corresponding to the maximum growth rate. And the map that you get is, of course, huge. It's 800, 900 reactions. So I'm gonna explain it to you by parts. Let me concentrate. Let me see. Let me concentrate first on the pentose. Let me concentrate first on the pentose phosphate pathway over here. You will see here on the left, these are fluxes for the wild type and the biodiesel producing strain. The fluxes are represented by the width of the arrow. They are the number of molecules per second to each of the reactions, normalized to the glucose input, so the entrance of glucose will always be 1. And there is all these small cofactor arrows here that are trying to represent where the cofactors are being created. So over here, there is NADPH being created, NADP being consumed. Here there's ATP being consumed, ADP being created, and so on. And the first thing that you see when you're comparing the wild type and the biodiesel producing strain is that there is a huge increase in the pentose phosphate pathway flux. Now, because pentose phosphate is the part of metabolism where you're creating most of the NADPH, that means there is a much higher NADPH production. So when I told this to Eric and Greg, they said, well, that makes sense, because in order to produce fatty acid ethyl ester, we need a lot of NADPH. And that's true, but from a qualitative point of view, but that's when these quantitative methods become important. So sure, you need more NADPH for producing fatty acid ethyl esters, but if you actually look at how much more you need and how much is being created, you see there is a mismatch there. So here, there is, it goes from 0.7 to 114, so it's like 80% increase. Now, if you look over here, the TCA cycle, which is where the precursors for the fatty acid ethyl ester are being produced, you will see that, yes, before in the wild type, you are having some flux that is going into fatty acids for producing membrane and so on. But the increase when you are producing fatty acid ethyl ester is just 1/6, 16%. And you were getting a much bigger increase before. Now, so the balance does not quite correlate well. Why is that? Because if you look carefully here, you will see that a lot of the NADPH is being created in the isocitrate dehydrogenase. And over here, the amount of flux that is being created, that it goes through the TCA cycle is much, much lower. It goes actually down by a factor of 50%, which correlates much better with the increase that we saw before. And why is that? Because we have much less TCA cycle flux, because a lot of the carbon is going into ethanol. So a lot of the carbon goes into ethanol. There is much less for the TCA cycle, then less NADPH is being created, and that is compensated by the NADPH creation in the glucose 6-phosphate dehydrogenase. We can see this even more clearly if you look at the NADPH balances. And the good thing of this method is that because we have all of the fluxes for the metabolism, we know where all of the cofactors are being created and consumed, so we can make this kind of graphs. This graph is all of the reactions that consume NADPH. I'm sorry, all of the reactions that produce NADPH, and these are all the reactions that consume NADPH. And this is for wild type and for the fatty acid ethyl ester producing strain. What you see here is that these isocitrate dehydrogenase and malic enzyme, which is the ones that we're talking about in this cycle, are creating up to 30 points in NADPH. And that goes down to 15, so it goes down by half. And the glucose 6-phosphate-dehydrogenase, that is part of the pentose phosphate pathway over here, is increasing by exactly that amount to compensate that. So it goes from 15 to 31, and this goes from 30 to 17. So one compensates the other. Now, well, once this system is in place, we can do this for all of the cofactors. I can do it for NADH, NADPH, ATP, ADP, AMP, all of them. And by looking at these graphs, you can find some interesting things. Like, for example, in this case, there seems to be a shift from in the respiration mechanism from dimethyl menaquinones into ubiquinones over here. Let me now go into limiting factors. One of the I'm trying. So my background is in theory. I was trained as a theoretical physicist, but I tried to make all of these mathematical methods available to the people doing experiments and trying to answer what they have as main questions. And one of the questions that I get all the time is, does my strain need acetyl-CoA or NADH or ATP? And basically, that's what I call a limiting factor. Now, the question that people are really making there is, if I have a reaction like the pyruvate dehydrogenase that creates acetyl-CoA, and I increase that, will I get more biodiesel or not? So if you take this reaction and you increase it by 10, 20, 30, 40, or 50%, am I going to get more biodiesel or not? And we can use the same methods, the MOMA method that I showed you before, to predict what is going to be the impact on biodiesel. Not only that, if acetyl-CoA was really a limiting factor, if you took the reactions that consume that acetyl-CoA and you decrease them by 10, 20, 30, 40, or 50%, you will free more of that acetyl-CoA to go into biodiesel. And this is exactly what we see. So hence we think that acetyl-CoA is a limiting factor in the case of fatty acid ethyl ester. Also, the interest of this method is that it gives you an idea of which reactions to overexpress or underexpress. So by looking at this graph, you can tell that these ones over here, are good candidates for either knocking them out or underexpressing them. And that's particularly valuable in JV because I may be doing all this more theoretical work. There are people in synthetic biology who are preparing, getting ready methods to take advantage of all of this. And one of them is this Mrs. Pacman for rapid strain prototyping, which is being developed in the Synthetic Biology Directorate. And the idea is that we will get, we'll be able to make knockouts in parallel. And in that way, by using my predictions and the Mrs. Pacman, we can try to fine-tune metabolism. As soon, well, if we can show that, well, we can do this reliably, and predictions are working fine. Finally, I'll speak very briefly about the knockout suggestions. I did produce some knockout suggestions to try to increase the biodiesel flux by using the MOMA principle that I spoke of before. This is the graph where I present the results, where here we have the biodiesel flux increase after you do the knockout, and this is the growth rate increase or decrease after the knockout. The kind of results you're looking for are over here, where you have a lot of biodiesel increase, but you don't take a huge hit in growth rate. Of course, the ideal results do not exist, so what we got is something in between. And I produced 3 of these knockout predictions in order to increase the biodiesel. Now, do they work? Well, at this moment, the growth rates that we're getting from these predictions are much lower than expected, which is one of the reasons that I'm developing the 13C metabolic flux analysis and flux balance analysis fusion, to try to get into better flux profiles from which to make predictions. And that is basically the main content of my talk. I will like to mention very briefly that we are working with yeast as well. We're doing 13C metabolic flux analysis for yeast, for bisabolin-producing yeast, and trying to increase the amount of flux into acetyl-CoA to try to increase the amount of bisabolin that's being created. And also, I am using 13C metabolic flux analysis for microbial communities, and I'm doing this for a system, the EBPR, Enhanced Biological Phosphorus Removal, that I did the metagenome for when I was at JGI, and that is mainly used for wastewater treatment. It's used for phosphorus removal. which is an interesting target because 80% of the biomass is one organism, a cumulolactic phosphatase. And so so this system is, it is almost like a single pure culture, but it has all of the complexity of a full ecosystem. And that's all I have for you today. My conclusions are what I explained before. The mission of host engineering is to direct as much carbon flux as possible into these high pathways. The two main techniques for flux analysis are flux balance analysis and 13C metabolic flux analysis, which I explained. I am working, and I just finished a method that will fuse all of the advantages of flux balance analysis and 13C metabolic flux analysis. And I showcase what are the possibilities of this method with a practical metabolic engineering project, which is this biodiesel-producing strain, in which we saw that the increased pentose phosphate flux is countering the decreased activity in the TSA cycle due to ethanol production. And also, I showed that acetyl-CoA is a limiting factor. And also that we're developing similar methods for both yeast and microbial communities. Let me just acknowledge the people that work with me in the quantitative modeling group, Jennifer Jean, and Weaver, and Chris Szymanski, and we work very closely with Vinay from Arkin's group. And there is also a variety of groups that we're working with in different organisms and biomolecules that we're interested in. And finally, Microbial Communities project is funded by LDRD and is in collaboration with Trina McMahon at Madison. The rest of the funding comes from DOE through JB. Thank you very much.
Is there a standard piece of software, or are you guys making your own software?
Okay, so there is a standard piece of software for flux balance analysis. There's a lot. There are some standard pieces of software for 13C metabolic flux analysis. There is Metran by Antonievich. There is 13C Flux by Vihert.
And it's built for E-COLI, or is it?
Well, many of these softwares, you just put the carbon transitions, you put by whatever reactions, and they can work out for yeast or E-COLI. But the field has advanced a lot in the last 2-3 years. So all of those packages are kind of obsolete, most of them. So there are many more interesting techniques that are coming out right there. So I have, for example, for doing this 13C metabolic flux analysis and flux balance analysis fusion, I had to develop my own methods to go into it. But I mean, basically, it's a very complicated optimization problem, nonlinear optimization problem, and that's scaling it becomes quite complicated.
So it seems to me too, I can't quite see through it, but you have to have something like a keg model underneath it, is that right? And then
So for flux balance analysis, there are some standard metabolic reconstructions, and there are people whose only job is just to do these metabolic reconstructions. Paulson did a lot of them. Many of them are available, but they used to be very artisanal procedures where people would spend, half a PhD just doing these metabolic reconstructions. Now there are ways to do at least part of these reconstructions automatically through something called seed and a few other methods out there. But manual interaction, manual creation is always necessary. For 13C, there's never been, until very recently, there's never been like a standard repository. People had their reactions with their carbon atom transitions, and it was very hard to compare one with another. It was very hard to take this model and try with this data. There was no standardization for that. Recently, Maranas, he started doing these metabolic reconstructions in an automated way. So he can do these carbon transitions, find these carbon transitions in an automated way for all the reactions in your metabolism. And he's making it public in a public database that should be coming very soon, like either now or next week or something like that.
And then these things get validated against, for example, the actual distribution of enrichment in the various positions of all molecules.
Typically not. That's the kind of things that we're trying to do now. So what I'm trying to do right now is add metabolic labeling for intracellular metabolites, for phosphoryl pyruvate, for all those.
So you're measuring 80, is that what you said?
I'm sorry, 80?
80 compounds when you do that?
We have 90 reactions. We are measuring right now like the 20 amino acids, and now the compounds we are measuring is the central carbon metabolism, glycolysis. We have, I think, like 10 or so. We are trying to increase that. There's people at JBA whose job is to try to get as much as possible from there. But one of the main problems with the whole field is validation. It started with the flux balance analysis. So you have these fluxes predicted by maximum growth rate. Who can tell you you're wrong? No one can, right? So the validation that they always had was, we can predict knockouts that actually make sense. But these are qualitative predictions, typically. So I'm thinking that once we have the flux balance analysis and the 13C all fused into one, then I can give you predictions for those biodiesel productions with error bars. And then we can make the measurements and see whether they are inside the error bar or not. So that's a way to validate it. The second one is to look at intracellular metabolite labeling. But yeah, one of the things that keeps me awake is, how do we validate these things? For a while I was doing 13C metabolic flux analysis, and I was telling Yingi, it's like, Yingi, no one can prove us wrong. I mean, really, no one can prove us wrong. I mean, we can make this assumption here, this ad hoc thing, or we can take this out, and no one can really prove us wrong. So that's one of the things that really worried me. And that's why I'm really, really insistent with the knockouts. Even though they are hard to actually make good predictions, it's really the only way we can show people like, yes, there is, this is holding water. So
it is all dependent on the steady state assumption?
that is a very important point too, yeah. This is something that we never make explicitly enough, and I should. And that's why I have it in the slide now, but I guess I should make it more obvious. So this slide here, oops, let's see. This slide here tells you exactly that. We are assuming steady state from the time that we feed this E-COLI all the way until we take the samples. We're assuming all the fluxes are in steady state here. Why? Because we need
For everything, not just for mainly
for fluxes. Everything, if metabolites are changing but fluxes are the same, in theory we don't care, as long as, I mean, the reason we do this is that these protogenic amino acids are going to be accumulated through all of this time. So you are assuming that we're in steady state and we're always having the same contribution from each time unit. When you're using intracellular metabolites, you don't have that assumption, because all of this, like PGI, F6P, and all that, their turnover time is much faster. So you are really seeing only what is being created at that time unit. Now, how long that turnover time is, that's something to be discussed. So in theory, looking at at the labeling patterns for these glycolysis intermediates will give us fluxes in real time instead of just the exponential. But that's research, and that's a research topic at this very moment. Mm-hmm.
So it seems like the growth rate in the production of a molecule of interest could sometimes be in conflict with one another.
Yes.
Are you assuming that at maximal growth rate, the maximum production of whatever molecule will occur?
No, I'm not assuming that. I'm using the MoMA principle, so which is, disputable. But basically, we're just assuming that it's going to be the closest compatible with the new constraints, with the new knockout. But I'm not assuming that. There are methods there are other methods to make predictions which are much better. So there are methods that will give you the knockout that will have the highest increase in biodiesel, maximizing growth rate at the same time. That's called opt-knock. And there is something called opt-force that will tell you exactly what are the fluxes that are compatible with your increased biodiesel production, let's say. But no, I'm not assuming that. Now, it's hard to have growth rate predictions which are accurate, though. Mm-hmm.
So there's been a lot of work on improving the predictions of growth by looking at regulatory effects.
Growth, no growth.
Yes.
Yes. So these are qualitative predictions.
Right. And so I was just wondering, do you think those regulatory effects would be
Yeah.
useful?
Yes.
Or do you think that you have enough constraints from your labeling that you don't need?
I think they will be very useful, very interesting. But I think that you get more bang for your buck by looking at metabolite labeling. So if I had to hire a full-time employee for either doing transcriptomics or metabolomics, I would go for metabolomics. That's my impression right now. But if I could have both, I love both, So, but Nathan Price has been showing that by having this transcriptional data involved, you can get much better growth predictions, not only qualitatively but also quantitatively. So it's something that I would like to include. But thank you very much.